Welcome to this video on microscopy and magnification, designed for your GCSE Biology revision. In this video, we will explain how light and electron microscopes function, and how to use magnification and size formulas for your calculations. We begin with a starter question. How do scientists view structures too small to be seen with the naked eye? And what information can a microscope provide about cells? Microscopes are essential for viewing structures invisible to the eye. They help scientists understand cell organization, function, and biological processes at the cellular level. Light microscopes use visible light and lenses to magnify specimens, creating clear, enlarged views widely used in schools and laboratories. Key components include the eyepiece and objective lenses. Total magnification is calculated by multiplying the magnification power of these two lenses together. Light microscopes form images by using light to illuminate a specimen, while the lenses work in tandem to magnify and focus the view for the observer. Light microscopes are ideal for observing cell structures in school labs, and can even allow for the study of living specimens in real time. Electron microscopes use beams of electrons and magnetic lenses instead of light, allowing scientists to see structures far too small for light microscopes. Higher resolution is the ability to distinguish between two close points. This is why electron microscopes reveal much finer structural detail than light versions. It is important to distinguish between magnification, the size of an image, and resolution, the ability to see detail within that image. In summary, while light microscopes are practical for classroom study and live specimens, electron microscopes provide superior magnification and resolution for detailed research. Finally, magnification is the ratio of an image's size to the object's actual size, usually written with an X symbol, and has no units. Actual size is the real dimension of the specimen being observed. Remember, 1 millimeter equals 1,000 micrometers. You can calculate the actual size by dividing the image size by the magnification. To perform GCSE Biology microscopy calculations, use this key formula. Magnification equals image size divided by actual size. Magnification is a ratio and has no units. The formula triangle is a handy tool to remember the relationships. Simply cover the variable you need to find, magnification, image size, or actual size, to reveal the correct equation. Units must always match before you calculate. Never divide millimeters by micrometers directly. Convert one value, so both use the same units throughout your working. To convert millimeters to micrometers, simply multiply by 1,000. For example, 2 millimeters is 2,000 micrometers. Always perform this conversion before using the magnification formula. Conversely, to convert from micrometers to millimeters, divide by 1,000. For instance, 500 micrometers is 0.5 millimeters. Consistency in your units is key for accurate results. Let's look at unit conversion examples. 2 millimeters becomes 2,000 micrometers, and 0.5 millimeters becomes 500 micrometers, in both cases by multiplying by 1,000. Here is a worked example. If the image size is 40 millimeters and the actual size is 0.2 millimeters, divide 40 by 0.2 to get a magnification of 200. To calculate actual size, use the rearranged formula. Actual size equals image size divided by magnification. With an image of 30 millimeters and 150 times magnification, the actual size is 0.2 millimeters. To find image size, multiply magnification by actual size. For example, with 400 times magnification and a 25 micrometer specimen, the image size is 10,000 micrometers, or 10 millimeters. For a complete calculation, first ensure units are identical. Converting 15 millimeters to 15,000 micrometers allows you to divide by 30 micrometers, resulting in 500 times magnification. You can measure cells directly from micrographs using a ruler or software to find the image size, then apply the magnification formula to determine their actual dimensions. To use a scale bar, divide the measured cell length by the measured scale bar length, then multiply by the value the scale bar represents. Keep your units consistent throughout. Let's apply the scale bar method. If a cell measures 24 millimeters and the scale bar measures 6 millimeters, you divide the cell length by the bar length, then multiply by the scale bar's stated value of 12 micrometers to find the actual size. In practical microscopy, you'll prepare your own slides. Accuracy in preparation is crucial. Focus carefully to ensure you capture clear, repeatable observations of cell structures. To prepare a slide, place a thin specimen on a glass slide, add a drop of mounting liquid, and lower the cover slip at an angle to avoid air bubbles. When focusing, start with the lowest power objective to locate the specimen. Once centered, Use the fine focus knob to sharpen the image before switching to higher magnifications. Staining is essential for identifying organelles. Iodine solution helps visualize plant cell components, while methylene blue is commonly used for animal cells to improve contrast. When recording your observations, draw exactly what you see using clear, single lines. Avoid artistic shading, and always include a title, labels, and the magnification or scale bar. Avoid common calculation errors by ensuring units match before substituting into formulas. Remember, 1 millimeter equals 1,000 micrometers, and magnification is a unitless ratio. Practice with exam-style questions. For instance, with an image size of 12 millimeters and an actual size of 30 micrometers, convert to matching units first to reach the correct magnification. Always remember these key tips. Magnification has no units, and always convert units before calculating to avoid mistakes in your final answer. To wrap up, consider these review questions. How do magnification and resolution differ? How does a scale bar work? And why do scientists prefer electron microscopes for detail?